Just for information.
Okay. At the outset, I'd like to thank the organizers, Professor Satyam, and I'm really kind of honored to speak on this occasion because, as you mentioned rightly, that as in Dr. Muralidharan has been my supervisor along with Professor Banerjee, and uh, because of Dr. Muralidharan's kind of approach and help and assistance in whatever way he has kind of provided me, that provided the basis for introducing me to this wonderful world of electron microscopy. And so I'm kind of indebted to him for all his support that he has provided me. So with that short kind of thanks to Dr. Muralidharan, I would like to kind of start off with my presentation. And as you can see that it is on imaging defects using electron microscopy. And this is like one topic which is really close to me and is very important in material science. And this was one of the topics which Dr. Muralidharan actually introduced me to. So that's why then I chose this topic and I'm going to speak on this. Even though it's a little bit basics, but then it is, and since I saw that it's many students would be attending, so I'm kind of kept the talk in that manner. Though it might be a little bit of overlap, or rather most of the information many of the speakers, or rather all the speakers would be knowing, but it's a little bit for the students as well. So
yeah, you can continue, but keep a track on the time. Okay, yes, so yeah. 25 minutes max. Yeah.
Yes. Thanks. So this is the outline of the presentation. I'll be talking on the how material characterization with some examples, and then come on to the different defects and the imaging using electron microscopy. So this so basically now we have heard from all the speakers about how the importance of processing, how the processing changes the property, and that property is being governed by the structure, or rather the microstructure. So now the characterization actually forms the central theme for all of them. Like we need to characterize the material, or if it's electronic material, so then how the device properties are getting changed based on the processing, and the processing in turn, how it is related to the structure. And similarly, even for structural materials, how the different kinds of grains or surfaces, which Dr. Ashutosh was talking about, then other people actually spoke about, how all those are being controlling the property. So this is on the characterization, the importance of characterization. So this is typically showing some examples of heterostructures, which are in the form of multi-layered. So what we actually need to check in those. So in order to characterize, we need to know about the individual layer thicknesses, the layer composition, interfacial roughness, and defects. And all these actually govern the final property or of the device that we're looking for. So there are some typical examples. This was one example which actually Professor Banerjee also showed. This is a gate and a 250 nanometer gallium arsenide indium gallium arsenide MESFET gate. So here, if we kind of see, it is very site specific, and we needed to understand about the layer and their layer interfacial roughness and the other things. So these we can only do using the TEM. Now, this is one example which I've just kind of picked up. Similarly, there are other examples. The first one is again showing the cross-section TEM of a space solar cell. Is one example. The another example where it is showing, this is again another example, which is a indium gallium nitride InGaN quantum well structure, and this is just the EDS map, the STEM EDS map. So using TEM, we can go beyond certain things which we could not have actually otherwise seen in a normal TEM, normal SEM or FIB based tomography. So we were able to use the FIB also. So in the site specific manner, in different orientations, we could pick up the samples and then characterize them, the cross section, as well as once we that this is kind of the analytical information of different elemental mapping and other things which we can get by EDS in this case, and even using PED in the TEM, we could get those. So I'll start off with one classic example, which is showing the processing property and the structure correlation and what's the importance of the TEM in that aspect. So this is one example, which in fact, this is part of my PhD work that I had done. So this is the substrate, and then we nitrate the substrate. And after that, we grow some low temperature gallium nitride, and then we anneal it. So this is where the small nuclei are then formed on the surface, the gallium nitride nuclei, and then we do the high temperature growth. Now, all the previous examples that I was showing, they're more or less lattice matched, and and the technology is pretty well developed that we don't see much of defects. But here, if you notice, this is the gallium nitride, and since we do not rather fabricate on lattice matched substrates, we do it on mismatched substrates, and due to a lot of differences in the lattice parameters, there are different stresses, and those in turn would lead to certain defects. And this is showing a weak beam dark field micrograph of the gallium nitride film, which shows multiple threading dislocations running right across the gallium nitride film. But this is the main thing to understand, that these defects in these films actually govern the properties. Like if we have, let's say, the LED that is shown here, if there are large number of defects, the efficiency of the LED reduces. Then if the second thing, if it is for the high electron mobility transistors, if there are larger number of defects, the efficiency reduces. So all that we need to control. The microstructure needs to be controlled, and for trying to do that, then we have tried to understand the evolution of these defects in this material, in gallium nitride especially here, and taking that as an example, I'm trying to show the imaging that we have done for these defects. So I'll start off with a simple example. Let's look at crystallographically as in what a line defect is and how we are trying to see it. And as an example, I've taken a sodium chloride crystal. There's the standard sodium chloride crystal. Now, this is the ball and stick model. Now, let's remove half of the planes of. So once we remove it, we see that in this particular orientation, we are able to see that there is a missing half plane. Now, let's say we rotate it about the vertical axis by 90 degrees, and we don't see. So it shows like a perfect crystal, and this would also come up as the perfect image if we are taking the TEM image. Similarly, if we do a 90 degrees rotation about the horizontal axis, still we don't see the defect. So this kind of shows one thing, that we are able to kind of image the line defect only when it is imaged along certain directions. So that single defect that we had created, this is a kind of a simulated or a theoretically created defect, we will be able to only image it when it is imaged along a certain direction. So this forms the basis for imaging conditions for dislocations. This is just for completeness. I'm trying to briefly explain the different modes of imaging. Now, the first one here is the diffraction mode, and where the diffraction pattern is the audience might be knowing here that as in the diffraction pattern is formed at the back focal plane of the objective lens. Now that diffraction pattern, from that diffraction pattern, we will be selectively selecting using the objective aperture the different diffracted beams, and that would then make up the basically the imaging vector with which we are imaging the different defects that I'm talking about. And this just for the completeness, the different types of dislocations, and based on the sense vector, we classify them as the edge and the screw dislocations. And for all these, like in the previous simulation that I was cartoon that I had shown, here we see that to match up the crystal structure, there are bending of planes, and that leads to the inherent strain. And that strain field is the actual one that is detected or observed during imaging in the electron microscopy. As an example, I'm showing this is the gallium nitride film grown on sapphire, and gallium nitride is known to have these three types of dislocations: the edge dislocations, which has a Burgess vector of 1/3 112 bar 0, the screw type dislocation, which is along the 0001 Burgess vector, and the mixed dislocations, which is a combination of both the screw and the edge. Now, what I had mentioned earlier regarding, so here this is the diffraction pattern that is shown. Now, if the image is done by tilting from the standard zone axis, or rather this orientation, to orientation which is taking a particular G vector, like here it is shown with the 0002, which is along the C. So what we see here are the C-type dislocations, or rather the screw dislocations in gallium nitride. And the imaging, when it is done using the transmitted beam in that orientation, it is showing me the bright field. And when it is by the diffracted beam, in which the dislocations are appearing brighter, and the rest of the, so this is a technique which we have used to kind of identify various types of dislocations and the imaging. We can image the dislocation. And this is actually leads us to the dislocation visibility, invisibility criteria. And using different G vectors, we are able to kind of identify what type of dislocation it is. So I'll take some example. In the this was something we were trying to analyze some gallium nitride islands and looking at the coalescence boundaries of the different islands. So site-specific samples needed to be actually prepared along at the interface of, or rather the coalescence boundaries. And this is one example of that. So here, what I've shown here is the sample when it is oriented along the 112 bar 0 direction. So now in that, and taking the, so basically after going to the zone axis, just slightly tilting to the different G vector conditions, the different two beam conditions, and imaging. So now we see that certain defects are visible in one and certain defects are not visible. And so using the visibility invisibility criteria, we know that those are like here what we are seeing, there are prismatic stacking faults are visible, the fault boundaries. And similarly in this, in a different orientation, we are able to see this with the G vector 112 bar 0, so which clearly shows that those are A-type dislocations. So we are able to classify the different dislocations that we are observing. And taking it forward, we can actually estimate the dislocation density, and which is very important for these kind of devices. If we need to characterize them and estimate the dislocation densities. So this is, we can take it forward to that. Then comes another technique. This is the weak beam dark field microscopy, with which you can get very clear in contrast between the defect and under different orientations. So this is showing an example where the same area is been imaged under different G vector conditions, or rather the different imaging vector conditions. And with this also we are able to kind of clearly see that the dislocation number 5 and 6 are pure A-type dislocations, and all the others are actually mixed dislocations, as the 5 and 6 are not visible with the G0002. Another way which we have actually looked at these dislocations are in the high resolution microscopy, and it is shown that it is being shown that as in, like, if we do the selective filtering using the specific G vectors in this SAD pattern that I had shown earlier, so then we are able to actually get a filtered map of the planes, and we'll clearly be able to identify the missing half planes. So that ways we have kind of looked at the different types of dislocations, the A, C, and A plus C type of dislocations in that particular area. And this is one example which kind of also shows that there is the disturbance in the C planes close to some steps which are formed. Other type of defect that we can actually see are the planar defects. Examples: twins, antiphase or inversion domain boundaries, and stacking faults. So now let's look at some stacking fault. Here in the first figure, what we see is when it is imaged under G101 bar 0 in gallium nitride, we observe that there is a stacking fault fringe that is present, whereas in the others it is not. And this is also kind of a visibility invisibility criteria for the different types of fault vectors. And we identify that this is a clear I2 type of a stacking fault in gallium nitride that we have observed. Using high resolution micrograph also we have tried to see. So here in this kind of mode, you can directly image the different stacking sequences, and we observe that there are certain alterations from the regular arrangement, and those then we can actually classify based on the different types of known stacking faults. We know that these are, in this case, the example that I'm pointing out, this is a example of 2 I1 type of defects that are observed. Then other types of planar defects that I was talking about, these are the twins. So this is again a gallium nitride. This is a cubic gallium nitride grown on sapphire, and what we observe here is that there are the gallium nitride different domains exist which are twin variants of each other. And this is evident from the FFTs with the common planes which are shown and which are mirrored, and the FFTs are mirrored along certain directions, and that is clearly evident. So these kind of defects we can directly observe using the high resolution TM micrographs as shown here. There is again another example, and this is the nitrided effect of nitridation. We had seen that the nitrided layer, which yesterday Professor Banerjee also talked about, that we identified the cubic nitrided layer. And the interesting thing to see is that the cubic nitrided layer, it forms as the twin variants, and we could clearly identify the two types of twin variants at that 1 nanometer interface nitrided layer. And that was also, the twin variant can also be identified from the this pseudo colored FFT that is shown here with the red in one and the green in the other, and which is also shown in the real filtered micrograph with the red and the green, the two different variants of the cubic nitrided layer. Another type of planar defect is the inversion domain. And so especially in gallium nitride, as in here, this as a fault vector which is along the C direction or the 0001 direction. So when we image this with the G0002, we see that there are vertical running faults, which is completely absent when we are looking at with the G112 bar 0. So that way, we know that these are some faults which are which have a fault vector which is along the or rather aligned with the 0001 vector. So then we went ahead and identified those to be as inversion domains based on the convergent beam electron diffraction technique, in which we have actually looked at the different disks acquired from the region A and region B, which is the inversion domain here, and we see that the intensities of the disk, say here, which is the 0002 bar, if we mark it as, the intensity becomes brighter in the other case. So, and that we have also gone ahead and confirmed with the STEM HUD-off micrograph, which shows that there is a clear shift in the gallium atom position at that very interface. Then I'll end up with this.
Yeah, Dr. Saha, another 2-3 minutes. So 1:30 we should close.
Yeah, I'm almost done. So so these are the point defects, and I'll end up with this point defects, that there are different kinds of point defects: vacancy defects, interstitial, extrinsic. Now these we can, these will be able to kind of identify if only using kind of the STEM HUD-off technique, where we'll be able to see the vacant positions or the different substitutions. So this is some work that I'm currently engaged in. This is showing magnetism in, I was trying to look at magnetism in molybdenum disulfide. So I've looked at the MOS2, and through other techniques of XPS and even STEM yields, we find that there is the presence of iron. And now if I look at this high resolution STEM HUD-off micrograph, we find that there are certain atom positions which are lower in intensity as compared to the rest of the matrix, or rather the molybdenum disulfide. And even the EDS map shows that there is a clear distribution of iron in the material. So this was an attempt to kind of dope the molybdenum disulfide to get room temperature ferromagnetic behavior in molybdenum disulfide, which is in general diamagnetic in nature. That's the thing, and I'll like to acknowledge firstly Dr. K. Muralidharan and Professor Dipankar Banerjee, who have supervised this work, and I'd like to thank past and present directors of DMRL for their encouragement, and all my other group members from Electron Microscopy and group head, and other friend of mine, Dr. Radhakrishna and Professor Vasu, who have helped in the gallium nitride growth IIC, whose results are in the gallium nitride characterization results that I have presented in this presentation. So with that, I'd like to thank once again the organizers for giving me the opportunity to speak on this occasion. Yeah. Thank you.
Yeah, thank you, Dr. Saha. So we are almost on schedule of the which has been behind schedule. So I must thank all the speakers, Dr. Balakrishna and Dr. Ashutosh Rath and Dr. Saha. And I would request Professor Satyam just to